Chris, you have a go.
Okay, it's 7:30 and I'm going to get started. Welcome to the June meeting of the Johnson Space Center Astronomical Society. I'm Chris Wells, Vice President of the Johnson Space Center Astronomical Society, and this is our agenda for the meeting. Our main presentation tonight is investigating the effects and changes of gravity in genetic regulation of human telomeres. The group TelOmG from the University of Toronto will be presenting. Next up, after that, there's the West Texas Star Party report. Chris Wells and Brandon Riddell and many other Johnson Space Center Astronomical Society members attended that event. So I'll be presenting that a bit later. And then last up, there'll be David Haviland with Star Party News. TelOmG is a group of undergraduate, graduate, and high school students from the University of Toronto. They designed a research experiment to study the effects of reduced gravity on human telomeres, inspired by the NASA's Twins study. The team has been selected to participate in the Canadian Reduced Gravity Experiment Design Challenge and will be flying their experiment on a parabolic flight this August. They aim to both further the understanding of how spaceflight impacts the human body and improve human health on Earth. TelOmG has been designing, building, and testing their experiment over the past few months and are very excited to share their work with you. With that, I'd like to turn it over to the team.
Hello, everyone. Thank you so much for joining us tonight. Our name is TelOmG, and we are a research group from Toronto, Canada. My name is Emma, and I'm an incoming third-year student at the University of Toronto studying aerospace engineering. Tonight, we'll be walking you through the work that we've done for the last 7 months and what we soon hope to achieve. We're currently investigating the effects of changes in gravity of the genetic regulation of human telomeres, and we'll be discussing our research, our design process, and where we are currently at with our work. Before we get started, here's a couple photos of our team. We are a team of 11 undergraduate, graduate, and high school students based at the University of Toronto, and our programs range from aerospace engineering to life sciences to biomedical engineering to physiology. So altogether, we've been able to combine our interests and our skills to complete such a multidisciplinary project. Every year, the Students for the Exploration and Development of Space, or SEDS as we like to call them, runs a competition in which undergraduate students can propose experiments that they'd like to see run aboard a parabolic flight. In other words, groups come together to pitch their ideas for experiments to be run in microgravity. Back in November, TelOmG was inspired by NASA's twin experiment, which you'll hear all about in coming slides, and came together to propose our own experiment, which was to explore the effects of microgravity on human telomeres. We were incredibly excited to be selected and have been working hard on our research ever since. Through SEDS, we have worked with experts from the Canadian Space Agency and the National Research Council of Canada, who provided advice on our design, our scientific experiment, and the outreach we've conducted, but more on that soon. But before we jump into an explanation of our research and our design, let's begin by explaining how we'll even experience microgravity. So, in a parabolic flight, the flight accelerates upwards at a steep angle, experiencing about 2 Gs of hypergravity as it pulls into the parabola. So that would be the little blue part that you see right at the beginning there. At the peak, everything within the plane is in free fall, so it allows everything to float as if it would in space. So that's that red area that you see in the middle. At the peak, we experience this period of microgravity, which occurs for about 20 seconds during each of these parabolas. This provides us with the opportunity to conduct microgravity research without ever having to go into space. So this August, we will be flying on board the National Research Council of Canada's Falcon 20, and we will be conducting data during 5 parabolas or 5 periods of microgravity. Next up, Dunya will be talking about our research topic and how we came to be.
Thank you, Emma. So before we delve into our experiment design, we're going to walk through our research topic and some background information that will help lay out the groundwork for our experiment. So our team was inspired by the NASA twin study, and this was a year-long biomedical investigation conducted by NASA to evaluate the health of identical twins, Scott and Mark Kelly, where Scott went to space while Mark remained on Earth. Given the identical genetic makeup of the twins, this study offered a very unique opportunity to investigate the long-term effects of spaceflight on human health. One of the most surprising results of the study was that Scott's telomeres lengthened in space, and it was surprising because telomeres shorten over our lifespan typically. It is unknown why Scott's telomeres lengthened, but what we do know is that there are several factors in spaceflight that might affect telomere dynamics, including microgravity and radiation. And this is where our project comes in, as we have a unique opportunity to isolate microgravity as a factor and investigate its effects. So the logical question that comes in next is, what are telomeres and why do we care about studying them? So the DNA in our cells is organized into chromosomes, and telomeres act as protective caps at the end of our chromosomes. When our cells replicate, the chromosomes get pulled apart to enter the new cells, and this process can really damage the chromosomes. That's why the telomeres are important. They prevent our chromosomes from damage as they get degraded in place of the chromosomes. As our cells divide, the telomeres will naturally get shorter and shorter, which means that there's less protection for the DNA. This translates to the fact that telomeres are a sign of aging. They're also associated with some chronic diseases and mortality, as well as risk of cancer. All of this together makes telomere dynamics super important to understand as we aim to improve human health. Next, Miranda will give us some more information on telomeres and microgravity.
So, as Dunya mentioned, telomeres typically shorten as we age, so exactly why did Scott Kelly's telomeres lengthen? Essentially, this could have been caused by two key factors involved in telomere activity, including telomerase and the Shelterin complex. Telomerase is a protein RNA complex that adds nucleotide repeats to the telomeres themselves, and is a process that is regulated by several proteins. Shelterin, as the name suggests, serves as a kind of shelter for the telomere that will protect it from DNA degradation or uncontrolled telomerase activity. Ultimately, two conditions affect telomerase and promote telomere lengthening. In terms of altered telomerase activity, this can be affected by regulators, access to binding sites, or the given conditions. Alternatively, changes to transcription of telomerase, regulators, and the Shelterin complex proteins can impact the length of telomeres through factors, including transcription factors, degree of methylation, and other environmental conditions and stressors. After reading about the NASA twin study, we looked at other papers to gather a better understanding of how telomeres might be affected in microgravity. Work by Teal, Wayland, and Narowski were fundamental to our curiosity and gave us insight on what we might expect from our cells in microgravity. Teal showed that short periods of microgravity contributed to alterations in gene transcription in both U937 and Jurkat cells. Wayland found that microgravity can induce epigenetic and methylation changes in genes. And lastly, Narowski showed that there was variability in gene expression at various time points throughout the flight. In our experiment, we want to look at how telomeres are affected by microgravity, and to do so, we will bring some cells on a parabolic flight. Since each parabola only lasts about 20 seconds, the cells won't really be in microgravity long enough to see actual changes in their telomere length, so instead we want to see how microgravity affects expression of certain telomere regulating genes. To do so, we basically want to freeze the cells before and after periods of microgravity, and we can do this by injecting the cell samples with a liquid called a lysis buffer. This ruptures the cell membrane, killing the cells and stopping metabolic activity, essentially freezing them in time. So if we lyse some cells at the beginning of microgravity and other cells right at the end of microgravity, we can see what changed in between. We have established two hypotheses that we intend to test in our own experiment. The first is that exposure to microgravity will cause transcription of telomerase and related proteins that will reflect positive regulation of telomeres. And the second is that microgravity exposure will cause an increase in methylation at the H-TERT promoter. Now I will pass it on to Mackenzie, who will talk more about the scientific processes of our experiment.
To overview our scientific plan, during this summer leading up to the flight, we'll be culturing a population of Jurkat cells, which are an immortalized human T cell line. We'll be doing preliminary testing to validate our cell lysis protocol and genetic extraction methods. Then, during the flight, we will execute the timed lysis of Jurkat cells between periods of microgravity to preserve the state of the cell's genetic material. Immediately after the flight, we'll be heading to the Pelling lab at the University of Ottawa to do our genetic extraction procedure to isolate RNA and DNA from the samples. and then we'll freeze those down and head back to Toronto. From there, we'll split into 2 tracks of investigation. Using the RNA, we'll convert to cDNA so that we can quantify the expression of each gene of interest using quantitative PCR. Using the DNA samples, we'll do bisulfite conversion so that we can run methylation analysis via methylation sequencing. Our preliminary testing can be broken down into a few different categories. First is a full genetic control. Using normal cell lysing procedures and conversion to cDNA, we'll practice our techniques, identify any areas of concern with our protocols, and importantly, assess the quality of our qPCR primers, because we'll probably be designing some custom primers for this project. Then we'll be testing our cell lysis in media. So typically culture media is removed from the cells before they're lysed, but due to the nature of our experiment where we need to keep the cells alive and happy until the moment of lysis, we won't be able to remove the media prior to lysing, and the lysis buffer will be somewhat diluted by the media upon injection. We'll also be assessing what happens when cell samples sit in this lysis solution for a couple of hours, as it's usually removed after a few minutes. We'll test different concentrations of lysis buffer in media solutions, as well as whether it's worthwhile to add a solution like RNA Protect to stabilize our samples and prevent degradation of the genetic material. Finally, we'll run a simulation of certain conditions of the flight campaign, removing cells from the incubator for a number of hours, exposing them to ambient gas compositions, different temperatures, and the long incubation in lysis buffer. We'll be assessing whether there are any changes in expression of our genes of interest, simply due to these experimental conditions alone, as we know that these might cause the cells some unusual stress. So next, Anthony will speak about our in-flight scientific procedures.
So following the preliminary testing, when we have all of our protocols, reagent volumes, stuff like that in order, we are going to execute this experiment and lysis cells in flight in August. So as mentioned, the flight consists of a number of parabolas and therefore allows us to collect data from a number of different time points. In total, we have 12. So firstly, we plan to lysis cells in flight prior to any of the parabolas, which is going to act as a baseline control for the following samples that are going to be collected under altered gravity. We will then collect data across the 5 parabolas, as mentioned, 1 sample before the period of microgravity and 1 after. That sample before acting as a control for the respective post-microgravity sample. So an additional benefit of 5 parabolas is we hope to see induced changes that might compound as the cells are exposed to more and more microgravity. Finally, we have 1 last sample point that's collected towards the end of flight, following all of the parabolas, and this is going to sort of act to see the effects of all flight and might serve to capture some delayed alterations to transcription or methylation that might have been induced by the changes in gravity. So at each of these time points, we plan to have 3 biological replicates, so 3 cell samples, and each of those cell samples is going to be 1 × 10⁶ cells. So I don't think the slide loaded for that one, so this is what I was referring to. Following the on-flight lysis, immediately post-flight, we plan to take those lyse samples and complete the remainder of all-prep DNA RNA extraction provided by QIAGEN. So after this extraction, we will have isolated DNA and RNA, in which we can perform downstream analysis. We will likely convert RNA to cDNA at the time of this extraction because cDNA is more stable, and then that can be frozen and we can perform analysis at later dates. So moving on to the analysis of the RNA samples, we are going to look at the transcription of a number of important genes, and we'll do so using QIAGEN's Quantinova CyberGreen qPCR method. So as per the specifics of the genes, we have a couple of controls or reference genes. So GAPDH is going to serve as our reference gene. This has been studied in Jurkat cells under microgravity, in which there is no changes observed. MT1X is going to serve as a positive control. So this, along with GAPDH, are unrelated to telomerase, but MT1X has been shown to have increased transcription in Jurkat cells under periods of microgravity. So getting into the genes related to telomeres and telomerase, The two components of telomerase are likely the most important we will be measuring, that being TERT, the enzymatic protein component, and TERC, which is the RNA template. We also have the 6 components of the sheltering complex, arguably the most important there being TERF1 and TERF2, which are the components that directly bind to telomeres. We also have some related proteins involved in the regulation of telomerase and telomere length. So DKC1 attaches to the enzymatic portion of telomerase and promotes its binding to telomeres. Tankerase inhibits shelterin binding to telomeres, and PINX1 promotes shelterin binding. So all of these proteins that are related to telomerase have shown or have previous studies where their transcription has been shown to be related to either telomerase activity or telomere length. So that was the criteria for choosing these and why we are analyzing them. Next, getting on to the DNA, we plan to look specifically at the methylation analysis of the TERT promoter. So we chose to focus here for a number of reasons. First of all, in the twin study, in the flight subject, the T cells had increased methylation comparing pre-flight to late flight samples. It's additionally been shown that microgravity can induce changes in methylation, and that associated changes in TERT methylation are related to its transcription. So that all fits together nicely to have some potentially important findings related to impacts to methylation because of microgravity, and therefore impacts on the transcription of TERT and telomere length. So this methylation analysis is an area that is relatively novel for all members on our team. So we're currently working on confirming the methodology for its assessment. The current plan is to perform bisulfite conversion of the DNA, which will convert all unmethylated cytosines to uracil in the sequence. And we then plan to perform PCR amplification on 2 distinct regions in the TERT promoter that have several methylation sites. After this expansion, all the unmethylated cytosines will now be represented at thymines, and there will be distinct differences in the sequence of unmethylated versus methylated TERT promoters. And those can be compared through sequencing methodologies like pyrosequencing or Sanger sequencing. It's worth noting that this process is known for being quite difficult, but luckily TERT methylation has a focus of study, particularly in cancer cells. So there's several protocols and methodologies developed, including primer sequencing for the TERT promoter that we can use. Finally, before I get into or pass it along for the engineering design, just want to go over some potential limitations with these scientific processes and some options for future work. So our experiment is conducted in Jurkat cells, but this is a cancerous cell line, and therefore there might be some impacts to the telomerase activity and telomere length of the cells. So a good follow-up would be to do this in a primary cell line, particularly in another T cell line, so it can be related back to the twin study. Next, increasing the scope of the experiment, either through RNA-seq or whole genome methylation analysis. This will allow us to look at other aspects unrelated to telomeres, where there could be other changes in transcription or methylation. Finally, more longer-term experiments so that telomerase activity, the protein levels of telomerase, and telomere length can be assessed are definitely warranted. But again, that would need longer-term microgravity for those changes to occur. I'll now pass it to Taylor for the engineering design.
To successfully perform our cell experiment in altered gravity while riding on a parabolic flight nonetheless comes many unique engineering design challenges and requires careful consideration of operational, material, and logistical constraints. Overall, the guiding design question we must answer is, how do we lyse cell samples at time points of interest in microgravity? By breaking this question down, we reveal that our experiment must maintain cell viability until lysis, which requires thermoregulation, gas exchange, nutrient access, and sterility of parts. It must store and transport lysis solution in microgravity, which requires a sealed lysis distributing subsystem. It must precisely control lysis injection into cell samples during parabolas, which requires electronic automation and it must operate within the constraints of Falcon 20, which has limited power supply, weight capacity, and physical space per experiment. Additionally, we must balance the feasibility, complexity, and assembly of our design with the access to resources we have as an undergraduate group, especially during a pandemic. As a self-funded group, the cost of design, experimentation, parts, and testing must be carefully considered to stay within our budget without sacrificing quality or capability. And lastly, we must prioritize designing for safety and reliability because of the variability in flight environments, because of pressure and temperature changes, turbulence, vibration, and stresses. And we have only one opportunity to perform our experiment during the flight campaign. On the next slide, Aaron will explain the 4 major steps of the experimental procedure.
Okay, I'm going to walk us through the main concepts before we get into the details of the design. So this slide only contains part of the apparatus. The blue container in the back contains a bag of lysing agent, which is the liquid that we need to inject into the cells to essentially freeze them in time. A syringe on the bottom left pulls lysis from the bag and then pushes it through some tubing into little bags that contain samples of cells. A 3-way solenoid valve, which you can see by the little yellow piece, controls when the lysis flows from the storage bag to the syringe and from the syringe towards the cells. Here you can see a metal rectangular flow distributor. This takes in lysis being pushed from the syringe and splits it to 12 different output ports. Each of these ports will be connected to a 2-way solenoid valve that will open to allow lysis to be pushed to the bags of cells. So an operator on board the parabolic flight will push a go button at each lysing time point of interest to begin the injection cycle, which we will go through now. So here you can see we're in state number 1. So at the beginning of a cycle, the syringe will be full of lysis. The 3-port solenoid valve will connect the syringe to the bag of lysis. Once the operator clicks the go button, the 3-port valve will switch to connect the syringe to the flow divider. One of the 12 2-port valves will open, allowing liquid to flow to 3 bags of cells. Now we're at state 2. So the motor will spin, compressing the syringe and pushing lysis through the tubing to the 3 bags of cells. Now the syringe has pushed all of the lysis into the bags of the cells and is compressed, so all fluid in the system is at rest. Next, the syringe needs to be refilled with lysis before restarting the cycle for a new set of cell bags. The 3-port valve will switch back to connect the syringe to the bag of lysis, and all of the 2-port valves will be closed. This brings us to step 4, so the motor will spin in the other direction, pulling the syringe open and pulling lysis from the storage bag into the syringe. Once the motor has finished traveling backwards, we are back at step 1 and ready to begin the cycle again. This cycle will repeat at each of the 12 time points of interest, lysing 3 different samples of cells each time. So now that you've seen the general lysing cycle, Luca is going to walk us through the detailed design.
Thank you, Aaron. So as you can see here, we have a 3D rendering of the physical infrastructure of our experiment, which we have chosen to divide into 2 distinct areas. On the top level will be all of our electronic components, which are quarterbacked by an Arduino Mega. The design decision to separate our electronic components from the rest of the experiment was made purposefully so that we could have easier access to them, as well as providing some informal barrier between them and the fluid area. Secondly, shown on the right, we have the fluid subsystem, which we'll go into more detail on soon. However, from a large-scale perspective, we have chosen to break the fluid subsystem down further into 3 further subsections, starting with the syringe, stepper, and reservoir subsection, the tubing subsection, and finally, the bagging subsection. Before looking further into the fluid section, these renders show how our entire experiment fits within a Pelican case, measuring 50 by 50 by 50 centimeters, which is required to fly as payload on the Falcon 20 aircraft. This case contains all our experiment throughout the flight and also provides power and data interfaces to the components. This case also must remain closed during the parabolic maneuvers, meaning our team members cannot access the experiment or lyse the cells manually themselves in flight. By satisfying this spatial constraint, we have created a design that would be easy to use in applications with considerable space constraints, which is often the case in aero or astro experiments. And again, before we get into detail about the fluid subsection, we want to discuss how we will be controlling the experiment. We have chosen to control our experiment manually using two push buttons, the green one that will act as a go button, and the red one which will act as an emergency stop button. We chose to incorporate the go button as a manual trigger rather than automating the lysis timing to avoid relying on sensors and risking our experiment firing at the wrong point in the maneuver or not firing at all. Flight crew on board the Falcon 20 will alert our experiment operators when we are going to enter and exit periods of microgravity, signaling the experiment operators to push the green go button, which begins the cycle Aaron just discussed. Furthermore, our entire design is self-sufficient save for an external power supply and the two push buttons. This decision was made to increase the versatility and self-sufficiency of the design, as rather than having to manually push a syringe or open a valve, we chose to automate these processes. Moreover, by using the high-density polyethylene as our shell material, we are able to plastic weld the walls together, as well as construct and indent the base. This design provides us with two primary advantages, those being easy accessibility and a leak-proof seal. As seen on the right, it is easy to access the electronic and fluid components. The floor walls and lid can simply be slid up and off, allowing for 360 degrees of access to the experiment. Once the experiment is primed and ready for flight, we can seal the joint between the floor and the walls, creating a watertight experiment, which is essential when working in micro and zero gravity. Now back to the fluid subsystem. Focusing first on the left, we have the stepper, syringe, and reservoir subsystem. As mentioned earlier, this is the system that will move all our lysis buffer and control the amount of buffer being pushed. In short, it acts similar to syringe pump, while being more cost efficient. Moreover, by including a reservoir, we have the capability to use larger amounts of lysis than would otherwise fit into a simple syringe. The main component which allows this all to happen is a three-way normally closed solenoid valve, which directs the flow and is controlled by the Arduino. Next, the tube subsystem shown on the right consists of a 1 to 12 manifold, as well as 12 two-way normally closed solenoid valves. This allows us to direct our flow effectively, as only one two-way solenoid valve will be open at a time. Thus, during step 2, when the lysis is pushed out of the syringe and into the manifold, by only having one solenoid open, the flow is forced into the trio of bags representing the current time point. Lastly, there is the bagging subsystem. This subsystem is utilized to split the lysis flow further into 3 more streams. Once past the single open two-way solenoid, the flow will be directed into this flow separator and then into our 7 milliliter bags, where our cell samples will be waiting, thus completing the movement of the lysis buffer. This will be repeated 12 times with a single different two-way solenoid opening at each sample point. Of course, since we are using cells, the bags which we chose to store them within was a complicated decision, which Taylor will now walk through.
A major design challenge was to interface cell cultures to the lysis distributing system without the risk of prematurely killing or contaminating cells. After much research, Origin Permalife cell culturing bags, shown on the left, were determined to meet our needs perfectly. The Origin cell culturing bags are gas permeable yet watertight and come with lure lock ports that remain sealed unless actuated. This self-sealing feature preserves the integrity of the cell bag environment both before and after experimentation occurs. The design of these cell bags has also given us flexibility in assembly because we can culture cells in advance, then connect cell bags via the lure lock ports to a primed and sterilized lysis subsystem on the day of flight. Additionally, the cell bags are non-rigid and expandable, which means injection of lysis solution and changes in pressure are easily accommodated. To store the lysing solution, another Origin bag, shown on the right, is utilized because of the simple lure lock interfacing and accommodation to changes in pressure and lysis volume as well. Ensuring viability of our cells also necessitates a tight control over the temperature they are exposed to. To avoid thermoregulating the entire experimental enclosure, we elected to create an insulated container that stores all 36 cell bags. The walls of the container provide insulation by sandwiching a layer of low infill percentage PLA with outer layers of high infill percentage PLA. A compartmentalized design reduces the power demand of heating, decreases the temperature response time of the system, and creates a buffer between the unregulated environment outside of the cell container and the cells themselves. Within the cell container, heating pads are placed above each row of cell bags to provide uniform heating when powered. A control loop will maintain a set point of 37 degrees Celsius plus or minus 1 degree Celsius by monitoring the enclosure's temperature with temperature sensors placed throughout.
So now we're going to talk about the electrical system of our experiment. So the experiment will be provided power through the Pelican case. The AC power supply on board the plane will be converted to 12 volts DC. A relay board will switch power on and off to different components. And the entire experiment will be controlled by an Arduino Mega 2560.
Collecting in-flight data is critical for demonstrating the intended experimental conditions took place in a controlled and consistent manner. This data will be used to help establish robustness of our results and, if necessary, help provide explanations for experimental errors or shortcomings. During flight, we will collect accelerometer data, temperature data, the injected lysis volume through lysis motor actuation distance, the time occurrence of each lysis event through the lysis button click, and a video of the entire experiment. In the end, all in-flight data will be displayed synchronously for demonstration and analytical purposes. As seen on the slide, the red lines indicate lysis button presses, which line up with experimental data at the corresponding flight environment data.
Looking forwards, the system we designed could provide useful in other harsh environments or on constrained research platforms. For example, it could be used for life science experiments on board future parabolic flights, on board the ISS, or in other highly constrained and challenging environments. Our results could be improved by improving the quality of data collected in flight. So even though we've incorporated accelerometers in our design, we plan to acquire and use the data collected by the Falcon 20's more accurate onboard accelerometer. Temperature data could be improved by adding more onboard temperature sensors. Although our cell sample bags are gas permeable, control of the cells could be improved by actively regulating carbon dioxide in flight. And additionally, lysing of the cells could be improved by adding an active cell and lysis mixing mechanism. So next up, Nicole is going to talk about our assembly, integration, and testing plan.
An important step of our design is to integrate the subsystems and to test how they work together. We have developed an assembly, integration, and testing plan to help us stay organized and on track with our design. We divided the plan into subsystems, such as the stepper motor, solenoid, and tubing subsystems. We planned and assembled subsystems separately and then begun integrating them together. We split the plan into phases to complete specific tasks in parallel so that items are ready to be integrated together. We are currently finishing phase 4 and starting phase 5 of our plan, which is the start of the integration of our subsystems. Next, Erin will talk more about prototyping.
So here I'm going to show you some of the prototyping we've completed so far. So we built a prototype of the whole system to begin putting things together and to complete electrical assembly and testing. You can see the syringe and the linear actuator on the floor of the assembly right in front. You can see the container that holds the bag of lysis on the left, and there are slots cut into it to access the tubing from the bag of lysis. On top of the lysis storage is the 3-port solenoid valve and the 12-way flow distributor. This leads to the 2-port valves, which then lead to the little cell culturing bags. Those little cell culturing bags are stored on a shelf, which we've built a prototype of the cell storage shelving, which will eventually be 3D printed. So we used these prototypes to complete some electrical testing. We successfully integrated the 2-port and 3-port solenoid valves, the syringe and the stepper motor, the flow distributor, the bag of lysis, the relay boards, the power splitting terminals, and the Arduino. We successfully completed a cycle of the experiment. So the stepper motor pulled the syringe, pulling liquid from a bag of lysis. The 3-port valve switched direction, and the motor then compressed the syringe, pushing liquid through one 2-port valve and into one of the cell culturing bags. Next, we need to incorporate the heating pads, the temperature sensors, the accelerometers, and the push buttons, as well as the flow separators. So we'll show you a video from this initial testing. So you should be able to see the motor pull the syringe handle back, fill it with some liquid, push it forwards, and then on the top left, you'll see the little cell bag fill up with liquid. Next, Sam is going to talk about our project timeline.
Our timeline and funding are important to keep our experiment running, and the outreach events allow us to share it with others. This is a quick overview of our timeline from our team formation to analyzing and presenting our results. Our team was selected for the CAN RGX competition in early December, and our experiment will fly on the National Research Council of Canada's Falcon 20 in August. Now Nicole will talk about our funding.
We have been sponsored by the University of Toronto's Division of Engineering Science, the University of Toronto's Institute of Biomedical Engineering, the University of Toronto's Engineering Society's School Endowment Fund. We have also received in-kind support from QIAGEN, Origin, Marlex Engineering, the Pelling Lab at the University of Toronto, and the NITS Lab at the University of Toronto. Next, Sam will talk about outreach.
Our public outreach events focus on youth interested in our experiment topics, including presentations to high school students, Girl Guides, and the Canadian Association of Girls in Science. We have presented at the Ascension 2021 conference in a virtual booth, as well as the 3-minute thesis competition. We also have 2 abstracts accepted for talks at the International Astronautical Congress 2021 and hope to publish in a peer-reviewed journal. It is also important to note our collaboration with the research labs listed that make our experiment possible.
And our team would like to thank our generous sponsors, including the University of Toronto's Division of Engineering Science, the University of Toronto's Institute of Biomedical Engineering, the Pelling Lab at the University of Ottawa, the University of Toronto's Engineering Society, the NITS Lab at the University of Toronto, Origin Biomedical, specifically Leanne Witt, Marlex Engineering, specifically Uwe Schabel, QIAGEN, and lastly, Shape Process Automation. Thank you all for listening, and you can leave any questions in the comments.
Go ahead, David.
Okay. First of all, I was thoroughly thrilled with this. As I mentioned in the rehearsal, biology is my day job, and actually I have grown both those cells, Jurkat's and U937 cells. I am curious, though, since this is somewhat of a transient study, you're only using, 4 or 5 parabolas to see if there's an early effect. Have you tried anything on the ground using some of those cell culture spinner vials to know if you might even if you have preliminary data that would suggest you might see something on a short-term effect? I mean, understand, I want to see this work for you. So I'm just wondering if you've managed to get a little bit of preliminary data as best you can to give you the indication that this is going to work when you actually take it up.
Yeah, so we haven't done anything on the ground with regards to the spinner flasks, but we did sort of start with Jurkat cells and pick this cell model because it has been used to look at transcription effects because of microgravity in the past. One of those Teal papers, I believe from 2018, assessed like a genome-wide transcription alterations from 20 seconds of microgravity. They did 20 seconds and 100 seconds, so there was some differences there, and that was a sort of a basis for choosing Jurkat as well.
Okay, that's good and encouraging. That hopefully you'll see something with the 5 or 6 parabolas then.
Yeah.
I'm also curious if there are jerkats and U937s that aren't going to be lysed, that will endure the 4 or 5 parabolas, and you'll check when they're on the ground for any kind of extended or long-term effects. I mean, analogous to steroid receptors, there may be some long-term changes, and I'd hate to see you guys miss those.
Yeah, so we're debating, like currently we have the one sample that is at the end. We're thinking of adding another one. So that one sample as of now is being lysed at the end of the flight and might see like prolonged changes that result from the microgravity. But you're right, it'd be very interesting to see a long-term one, especially because the NASA twin study saw the telomere length revert back to normal post-flight. So it would be cool to see some changes and then it go back to normal after as well.
Okay. What I do here in Houston is, and you guys are probably familiar with the technology, I run a flow cytometry core. Is there any possible extensions to this where you guys might be looking at possible change in surface markers as a function of gravity, microgravity, long-term or short-term?
Yeah, that'd be very interesting. I'm not sure about, just because of my knowledge, I'm not sure about surface markers relating to telomeres, but even microgravity as a whole, seeing effects on like other processes would be really cool.
It's unrelated to what you guys are doing, but we have found, depending on how jerkats and U937s are actually sorted on sorters, the higher the pressure, the more rattled the cells are, and the slower they are to resume cell cycle division. So there are impacts and things that if a sorter can impact a cell. For the audience, let me describe the process of a sort. It's basically like you going through a joyride at a theme park. It's going to spin you upside down, and it's going to electrostatically shock you, and it's going to throw you into a swimming pool. And some cells get along with that, and some cells don't. But the point being, whether it's that or microgravity, you will get gene changes. So I was just curious if the microgravity may alter genes as well.
Mm-hmm.
The other question, I may have missed the point, media and pH control. Somebody mentioned CO2 regulation. How are you having the cells buffered during this process? Is it say HEPIs, or are you doing carbon dioxide buffering, or what?
There is no carbon dioxide buffering. That's sort of a shortcoming of our experiment. The cells are suspended in, I believe it's in RPMI-based media. And again, sort of really looking up to Teal in her papers, but they looked at the jerkat cells and they didn't have CO2 regulation as well, just temperature in the same RPMI media. So we're again sort of basing off that.
If it's fully sealed, as you show in the bag, and there's no air, then there's no place for the CO2 to go. So your carbonic buffering will work just fine, unless you do something like heaps, HEPIs or heaps buffer.
We also chose the bags that contain these cultured cells are gas permeable, so they allow for gas exchange throughout the flight. We're just not actively regulating the CO2.
Okay. So you just have to watch and make sure they don't go purple on you in the process.
Yes.
Okay, I've hogged enough. Anybody else have questions? By all means, please jump in.
Okay, it looks like we have a couple that came in from the chat. The first one is, what are you hoping will happen with the results from your study? Any real-world applications? Any improvements with healthcare specifically?
I mean, I'm not sure about healthcare specifically, but for real-world applications, I mean, this may impact like long-term space travel in the future, specifically where there'd be long periods of microgravity or zero gravity, and impacts to telomere length is going to be vital to assessing human health up there. So that was really a big inspiration for it.
We also know that telomere length is indicative of chronic diseases and illnesses and health here on Earth. So if we can find interesting impacts of any factors, or even narrow down what affected the telomere length in the twin study, it could benefit human health here on Earth as well.
Okay, it looks like we have one more here, and this is looks like it's aimed at the actual experiment itself. Is there any feedback that the green or red buttons have successfully completed the actions you believe it should during the experiment?
Yeah, so we've individually connected the buttons to the Arduino and tested that it can read their inputs. We haven't integrated that with the lysing cycle, but we know that the buttons are read properly, and then if the Arduino has that input, it can execute one lysing cycle as we've tested. If you in-flight feedback, we'll have LEDs showing when lysing cycles are active so that we know whether the button input was read.
It looks like there's one more late one that came in. Any reason why Mark's telomeres got longer in space rather than shorter?
We don't really know, and then that's a big inspiration for the study and trying to figure that out. So that's why we're looking at transcription of proteins that would increase telomere length, and we can maybe associate that to the increased telomere length observed there, as well as the methylation, which in turn would affect transcription and then the telomere length, hopefully.
All right, I'm going to send it back to Chris. Chris, you're on.
Telomere group, that was an excellent team presentation. Thank you so much. Some fascinating stuff there, and raw research like this into this area is who knows where that's going to lead us, especially with all our space programs and our almost visionary plans in the future. So next up, I'm going to be presenting about the West Tex Star Party that was recently, some of us recently attended in May. It was conducted at the Prude Ranch in Fort Davis. So many of you know that TSP was actually canceled last year, and it was canceled for in-person this year, but it's actually online. They're having an online TSP this year. But TSP is generally an event that has around 500 or so people attend. And so unfortunately, due to COVID, it was canceled. But there's a whittled down or a much smaller event that's been conducted at the same place, which is the Prude Ranch in the Fort Davis area. So this Star Party was really in lieu of TSP. And as I say, a number of us from JSC AS attended. So first of all, West Texas. I love West Texas. I just love the beauty of the area. It's absolutely incredible. You can see me there being very happy in my element, being away from the Houston area and actually being just out there. I just love it. This is from a ridge just outside of the Prude Ranch. I said location. This is where it's all really happening. Up here, you've got the McDonald Observatory complex. You've got some other private observatories around here. And down here is the Prude Ranch, just above my head. You can start to see all these little white symbols there. Those are actually telescopes from the West Tex Star Party. This year, it was attended by about 60 people. As I said, it's a much smaller affair. But nonetheless, it was great to get all our equipment out there. And this, I think, slide represents that. Here's another view from the other side of the Prude Ranch. Once again, observatories are up there. I think that might be the Hobby Eberly telescope, if I'm not mistaken. Once again, just love the beauty and landscape around the Prude Ranch. But it also shows you that just by walking up to the ridge from the Prude Ranch, you actually get within distance of seeing the research grade telescopes out there on the ridge. So it's all very close together, and just a great place to actually take your telescope and have a lot of fun. Here's another view. Just can't get enough of the views in that region of Texas. Absolutely love it. Can't speak too highly about it. So here's some other views for those who aren't into scenery views. Here's some views of some telescope equipment. I attended there mainly with a number of JSC AS people who attended. The Millers were there. Brandon was there too. This is Brandon's scope. He has a sort of an 8-inch, I think it was f3.9 imaging Newtonian scope. I took on a number of telescopes with me. Those 3 I took with me. Some of them they're all old, some of them are borrowed, and there are none that are new. So this is kind of a collection of different equipment, some of it borrowed that I took just to kind of experiment and do some astrophotography there. The biggest telescope I have here is just a 5-inch refractor, and there's a sort of small 90mm refractor here. Also very good at good at imaging. So see, lots and lots of wires, lots and lots of equipment, getting everything working. Okay, some numbers. This is just meant to be a flyby about the trip. Some numbers. Where is the Prude Ranch? Well, it's about 630 miles from where I live in League City, in the sort of Houston area. So it's 650 miles away. And I'm practically, because I'm in Galveston County, I'm practically at sea level. So the Prude Ranch itself is 5,000 feet above where I'm actually sitting and presenting from now. If you go up to the ridge where you saw the McDonald Observatory, that takes you up to 6,790 feet. Here's some other pictures. These were taken by Brandon of the main McDonald Observatory. And the telescope in there, I think, is about 107 inches telescope, if I'm remembering rightly. Also, Brandon shared a picture of here the VLBA, or the Very Long Baseline Array radio telescope. He gave me a picture as well of the picture outside of the gate of this large telescope, where it said it's a continent-sized telescope. There are basically 10 of these across the U.S. that enable you to take very, very fine resolution radio telescope images. Some other numbers. We actually took a hike. It wasn't just about astronomy this trip. It was also about hiking as well. Here's Brandon's on the right. There's me on the left. And this is us climbing up to Guadalupe Peak, which is the highest point in Texas at 8,751 feet. It's about a 2.5-mile drive from Prude Ranch. And once you do the 2.5-mile drive, you've got another 8.4-mile hike to go up to the top and back. And that just there shows me that I did it and can still do it. So that's good news. Any trip has some drama. So we had some drama on the trip. This is the observing field here. And we actually had a herd of goats come through the telescope observing field. That was kind of fun and unusual to see. They were all escorted by, I think, 3 different dogs, sort of sheep dogs, goat dogs, whatever you call them. And this went, so that was some drama there. That's one of the dogs laying outside of and staying guard on one of the, actually it's Marty's telescope tent. And here's another picture of some drama. We actually had some dust devil activity that went straight through and ripped off the cover of Brandon's telescope. So that was kind of interesting to see as well, a dust devil come through. We also had an incapacitated vehicle issue when we were there, and this is Brandon's vehicle, and this is my truck. So if you remember, I said I'm 630 miles away from the Prude Ranch. Ended up towing the vehicle all the way back, and the guys there had a bit of fun with me and put a label on the side of my vehicle saying Limey Auto Transport. So I thought I'd take the joke on that one. And lastly, here's just some astro images taken of that trip. I'm still processing images. And of course, you'll recognize some of them off the bat here. The Pinwheel Galaxy and Ursa Major on the left. And on the right there, you see Omega Centauri and Saturus and a bunch of other things like the Pinwheel Galaxy here. And that concludes my presentation. Is there any questions?
I think you're good, Chris. I'm going to hand it over to David. All right, David, you're up.
All right, let me make sure that I haven't muted my own mic. All right, Star Party News, Chris. That looked fun. I need to get my way out there at some point. All right, Star Party News for June. anytime computer. All right. On May 21st, we had an event at the Hack Winery. I will talk about that. On June 10th through 12th, there was a virtual. There is still the virtual Texas Star Party. Part of it's going on tonight, and there's one more night tomorrow. It started last night. FBAC took part in a bit of the role on that. Coming up, July 17th, we have an event at the Hack. September 17th. So far, nothing has cropped up in the calendar yet. October 7th through 9th would be our club's trip to Fort McCavitt. October 15th will be the all clubs. They're still planning on having that on October 16th. If things still keep relaxing, we hope to have some form of the Houston Astronomy Day at the George Observatory. November 1st through 6th, El Dorado, just where Chris just talked about. There'll be the El Dorado Star Party there. And then the moment we finish the year up on November 19th with an event at the Hack Winery. The event on the 21st was, sorry, no pictures. The event was totally clouded out and somewhat drizzly, as the forecast, in fact, suggested. Despite having 219 RSVPs, there might have been 40 people in attendance, with a couple of folks asking questions, because I brought my Dob, and Connie and I brought the Dob, and Bob and Leslie Eaton brought brought one of their scopes and a pair of binoculars, and it was enough to attract a little bit of a crowd. We did hand out a few flyers for the club. We pretty much wrapped up everything else, and we were on our way back home by about 8 p.m. because there was not anything to see. At the moment, we can't win for losing when it comes to weather these days, particularly given the weather for the last 5 months. Texas Star Party is going on now. If you catch up with the Facebook on Texas Star Party, you can see what's going on. So I thought I'd throw that in. There's one more night tomorrow, unless you want to catch it off the hands here. It is going to be lots of it are being pre-recorded. I know Larry Mitchell, the segment that FBAC did last night, David Levy's, and Steve O'Meara were all pre-recorded. So this will eventually be put out for people to review again. So it's their first attempt doing this. And if the last two, tonight and tomorrow night, are going to do anywhere near as well as last night, it's going to come off really, really slick. And with things lighting up, hopefully we will get back to actually doing in-person outreach. And then this one is an old one. It's actually, this is a couple of times I've found this. It's turned out to be somewhat true. You've got Calvin and Hobbes looking into the heavens. And Calvin and Hobbes comic from 1992 referred to the start of the universe as the horrendous space kablooey, saying that Big Bang was too boring. The term horrendous space kablooey has actually since become popular among scientists and has found its way into textbooks and university courses. So there's a little bit of trivia you may not have known. All right, that's it. And back to you, Chris.
Okay, Chris, you're up.
Excellent, David. Thank you so much. Once again, thanks again to TelOmG for their excellent group presentation. Next month, our next meeting is on July the 9th. We have Aaron Clevenson from North Houston Astronomy Club presenting on Galileo's toes. So do come back and join us then on July the 9th. How can you not want to with a title like that? Thank you ever so much for joining us this evening.